We'll see. Okay. Good afternoon. Good afternoon. Today we will continue our lecture about Down syndrome, which was the last lecture and the first lecture of clinical genetics. And next, I will start the lecture about genome imprinting and also uniparental disomy. Okay, and Down syndrome. Do you have any questions about Down syndrome or last lecture?
No.
Can you hear me? Okay. Can you see the presentation? Yes?
Yes.
Yes.
Okay. Okay. So we continue. Phenotype of Down syndrome, and here you can see what are the compounds of this phenotype. Congenital malformations, I mean heart defects, mainly heart defects in about half of children with Down syndrome. Gastrointestinal defects, only in some percent of Down syndrome children. Hernias, different hernias in about 20 to 40% of people with Down syndrome. Thyroid insufficiency. It is present in, it may be present in almost all children with Down syndrome. And we should, even if it is not present in a newborn with Down syndrome or infant with Down syndrome, we should always examine a child with Down syndrome for thyroid insufficiency. Yes, because it may occur in every moment of the life. Okay, so we should examine TSH, yes, and many children with Down syndrome should be treated for thyroid insufficiency, and it is about 50% of children which are treated for thyroid insufficiency, children with Down syndrome. And sex development disorders, cryptorchidismus in almost every boy with Down syndrome, infertility in adult men with Down syndrome. The women are fertile. The women with Down syndrome are fertile. They have a risk of having, of course, they have a risk of having children with Down syndrome. How high may be this risk? What do you think about it? The risk of having a child with Down syndrome for a woman with trisomy, simple trisomy 21. If her partner is healthy. What do you think about it?
Low risk.
50.
50%, yes, of course, 50%. So this woman may have half of children with Down syndrome, half of children without Down syndrome, healthy children also. Okay. And what else about the phenotype? Loss of hearing, ophthalmic disorders. We should examine it. This, sorry. We should examine hearing and vision in children always, because it is very important for their intellectual development also. Yes, and there may be neurological disorders like muscular hypotony and also seizures. And many children have sleep disorders. About 50% of children have sleep disorders. Some of them have dyspnoea, and many children, almost all children with Down syndrome, have teeth disorders or parodontosis and so on. Okay.
Why?
Why?
Why
I don't know.
specifically teeth disorders?
Why these disorders? I don't know. It is maybe there is a gene on chromosome 21 which is the cause of these disorders. There are, for example, in some children maybe hypodontia, yes, too low number of teeth. In some children, the positions of these are not good. I don't know why directly. It's not. It is not directly linked to a gene or a part of gene of a mutation of a gene. Yes. So I can't mutation or additional copy of a gene which is located on chromosome 21. So I cannot explain you the mechanism of this. Okay.
Okay.
Maybe sometimes it may be also sometimes hygienic problems. I don't know, but in general, many children, even without hygienic problems, have disorders of this. Progeria. I have taught you that that it means accelerated aging, and because of what? Accelerated shortening of telomeres, ends of chromosomes, yes, disorders of DNA repair in many persons with Down syndrome, and additional copy of APP gene on chromosome 21. APP, which means amyloid precursor protein, the gene for amyloid precursor protein. Yes, and when there are additional copies of APP, protein APP, there are also in central nervous system additional copies of amyloid, which is stored in neurons. Okay. And what about Alzheimer's disease in Down syndrome? There is an accumulation of beta amyloid 42 in neurons because of this additional doses of APP, and also because of additional doses of gene which is called DIRK1, and also it is called Down syndrome gene. Yes, and it is located in this critical region of Down syndrome, 21q22 something. Yes, okay. And this gene, the protein product of this gene, inhibits neprilysin, which is a main factor degrading beta amyloid in the brain. It is a repetition. I would like to repeat it for you. Sorry. So, additional doses of DIRK1 inhibits neprilysin, And because of this inhibition, beta amyloid 42 is not degraded. Okay, it's another problem. Okay, phenotype: low unspecific immunity is unspecific immunity is very low, and low specific immunity is also very low. Low cellular and low humoral immunity because of low activity of T lymphocytes and B lymphocytes. And what about unspecific immunity? There is a additional copy of a gene which is called SOD1, SOD1, which codes for superoxide dismutase. Yes, and so as a result, we have a higher SOD1 activity, and this SOD1 enzyme eliminates ROSes, reactive oxygen species, produced by phagocytic cells. It is a weapon of these cells against bacteria. Yes, so phagocytic cells have low levels of this reactive oxygen species. Okay, and another mechanism, high amount of S100 beta protein because of additional copy of the gene for this protein on chromosome 21. Yes, and as a result, there is a higher binding of zinc. Yes, because protein S beta is a transporter of zinc, and higher zinc binding means lower activity of thymus and lower chemotaxis of neutrophils. Okay, you understand? Okay. Okay. And there is also a higher susceptibility to leukemia, like leukemias in general, which means mainly acute lymphoblastic leukemia, the main type of leukemia in children, acute lymphoblastic leukemia, ALL. So children with Down syndrome are 20 to 40 times more susceptible to this type of leukemia. Okay, but they are also susceptible to some types of myeloblastic leukemia. The main type is a very rare type of myeloblastic leukemia, which is called acute megakardioblastic leukemia, AMKL. And to this type, children with Down syndrome are 400 times more susceptible than children without trisomy 21. Okay. Normally, children almost don't have this type of leukemia. This is an adult type of leukemia. And children with Down syndrome are also more susceptible to cytostatic treatment because of different metabolism of folates, and also additional copies of, for example, beta-cystathionin synthase, which augment the activation, which makes higher the activation of cytosine arabinoside, ARA-C. Yes, so this cytostatic acts longer than normally. This its activity is longer than normally. And there are also many other mechanisms of higher susceptibility to cytostatic treatment. And many years ago, children with Down syndrome, which had leukemia, died not because of leukemia, but because of cytostatic treatment, because they were very susceptible to this treatment. Now the treatment of children with Down syndrome and leukemia is totally different from the treatment of 20th century, for example, yes, and also from the treatment of children without trisomy 21 but with leukemia, yes, because the children without trisomy 21 are not so susceptible to cytostatic treatment. They may accept very high doses of cytostatics, but children with Down syndrome cannot accept these high doses of cytostatic drugs. Okay. And another problem, organ cancers. There is a lower susceptibility to organ cancer in Down syndrome children, with one exception, which is testicular cancer. Questions? Questions, comments?
No.
No. Okay. No. And we can differentiate. We have many types of phenotype. Yes, many features of which are included in phenotype. Phenotype consists also of so-called behavioral phenotype, and we know that children and adult persons with Down syndrome are empathic, are friendly, they trust people, but they are stubborn also. Children and adult people in general. And many of people with Down syndrome have many skills. The main skills are music, dance, and so on. And sometimes design, something like this. Treatment and rehabilitation. There are no causative treatment of trisomy 21. Yes, there is only symptomatic treatment. We treat only the symptoms. There is no cure for Down syndrome, of course. Yes. What is the most important? The most important is early developmental rehabilitation, which includes also early logopedic rehabilitation. Children with Down syndrome should start logopedic rehabilitation before they start to speak. Yes, before, because the whole apparatus of speaking should be prepared to speaking. Okay. And what else about treatment? There are many clinical trials which deals with activation or inactivation of metabolic pathways which are changed in Down syndrome. Down syndrome means trisomy 21. Trisomy 21 means 3 copies of about 400 genes. Yes, so we have additional 400 copies of genes. Yes, and every additional copy means a change in at least one metabolic pathway. Yes, so these trials of with inactivation or activation of metabolic pathways are very promising. Yes, promising. And what about the future? Maybe we will have a causative treatment for Down syndrome and other trisomic syndromes. Maybe yes, maybe not. Maybe we will have tools for blocking of third chromosome 21. Maybe yes, maybe no. Blocking of maybe it is all possible. I think that in 10 years, 10 to 20 years, we will have some possibilities of treatment of Down syndrome. But I should remind you that treatment doesn't mean cure. We will treat more effectively people with Down syndrome, but it doesn't mean that that it will be possible to completely cure them. Okay, so they will have always Down syndrome, but maybe their intellectual functions will be ameliorated. Maybe the intestinal functions will be better, and so on, so on. And what about today's treatment? Symptomatic treatment. Children with Down syndrome should undergo many consultations, which means endocrinologic consultation because of thyroid insufficiency, ophthalmologic because many children, even up to 50% of children have ophthalmologic problems, otolaryngologic consultation because of hearing problems, and also because of larynx disorders, because of larynx hypotony. In general, children with Down syndrome have muscular hypotony. This hypotony concerns not only the muscles, but also, for example, larynx and palate and so on. Neurologic problems, for example, hypotony, general hypotony, cardiologic because of heart defects, orthopedic problems, orthopedic consultations. The first orthopedic consultation should be, as for every child, at birth. Yes, but later on we should do orthopedic consultations because of the possibility of instability of cervical vertebrae. Yes, because instability of these vertebrae may cause, for example, accidents during hypotherapy or during surgical operations. Yes. And what else? Sometimes not not obligatory hematologic consultations. Why? Because of the possibility of transient abnormal myelopoiesis, or because of leukemia. And what should be regularly verified? Blood cell count, and blood preparations, and so on. Yes. We should verify it every year, or it would be better every half a year. Okay. Questions?
No.
No questions. Diet. What about diet? In general, people with Down syndrome are obese. Yes, have a tendency to obesity. So they should be on low calories diet. Yes, and now many people believe in diet supplements. Sometimes some supplements it's not it may be good, but not many supplements. The only one diet supplement which is verified, it's green tea. The substances which are in green tea, not other supplements. Sometimes some compounds, micro compounds, some magnesium, zinc, and so on, so on. Yes, but not many supplements, not multivitamin preparation, not multivitaminization. Okay, only vitamin which are indispensable for a particular child. Okay. What is important for people with Down syndrome? Social integration, not only familial life, which is very important, but also social integration. Integration is the whole society. Yes. What does it mean? Acceptation, backup, financial from the government, yes, and also psychological from pedagogues, psychologists, teachers, and so on. Many people, yes. There should be well-developed children care for children with many genetic syndromes and disorders, not only for Down syndrome children. And also, governments should develop adult care, the centers of care for adults with genetic syndromes. Okay. There should be the possibilities of work activities for young people with genetic syndromes, for adult people with genetic syndrome. In general, people with genetic syndrome are equal to healthy people, yes? So they should have the occasions to participate in events of social life, in general in social life, yes? And all governments should develop so-called protected flats for people with different genetic syndromes. What does it mean? Protected flats, flats which have which have some rooms for these people with genetic syndromes, and 1 or 2 caretakers. Yes, not family. Family is always welcome. Yes, but caretakers, professional caretakers, who can help the ill people in administration problems and so on. Okay, and the children with Down syndrome should have many activities, artistic activities also, because they like it. Okay, social integration, participation in important events, social events like this woman. And the basic question, we know that there is an European Union project, Rare Diseases. It is a very important project. It has started at in this century, yes, but it is on rare diseases. Does Down syndrome is a rare disease or not? Rare diseases or orphan diseases, because nobody is interested in them. So orphan. These are diseases which frequency is 1 to 2,000. Yes, they are mainly in 80%. They are genetic or diseases or syndromes, and some 10% rare cancers and other diseases. Yes, okay. So, does Down syndrome fulfill the criteria for a rare disease? Yes or not?
No.
No. Why?
I don't know about the statistics, but I think there's a lot of people that has Down syndrome, and if they don't have it, some people are more sick, not more sick. It's wrong to say sick, but more than others.
Yes, and the general frequency of Down syndrome is higher than 1 to 2,000. Yes, it is about 1 to 700. So Down syndrome doesn't fulfill the criteria for rare diseases. Okay, but it should be included in this problem, because we have many other syndromes, yes, which fulfill criteria for rare diseases. Only children with Down syndrome are expulsed from this group of other syndromes. I think that it's not good for people with Down syndrome, because people with rare diseases have some privileges, some additional should have some additional money from a government or given to governments for make better their life, and so on, so on. So I understand that Down syndrome fulfill other criteria of rare diseases without the criterion of frequency. Yes. So it's not an orphan disease. It's too frequent syndrome. Too frequent syndrome. how much frequent? You can see here. Almost all people here have Down syndrome. Okay, and here you can watch an actress which who have Down syndrome. She is 20 years old, about 20 years old. She's a very good actress, even if she has Down syndrome. Okay. Thank you. It was the first part of our lecture, our common lecture, and now something about imprinting, genetic imprinting. Do you have any questions or comments and so on?
No.
No. Okay. So, genome imprinting and uniparental disomy is it is the topic of our second lecture. What does it mean, genome or genomic imprinting? It is a synonym for parental imprinting. We can tell this phenomenon as genome imprinting or parental imprinting. And what does it mean? It is an event and phenomenon which is totally physiological mechanism which is present in all higher eukaryota. Yes. It is an epigenetic event, so it does not consist in changes of genetic code. It takes place after transcription and translation. After transcription and translation. It only changes the expression of genes by, for example, blocking some particles in these genes, in the DNA of these genes. It consists in gene expression modification, but mainly a reduction of this expression. The main mechanism of genome imprinting is methylation of genes. Yes, methylation of genes, which means linking with methyl groups. Yes, and by this mechanism, the blocking of gene activity, of gene expression, silencing of genes. Okay. Other mechanisms are acetylation and deacetylation. And deacetylation causes the silencing of genes. Methylation or deacetylation. But the main mechanism is methylation. And when this gene methylation appears, it appears in late gametogenesis, and it lasts until a very early period of development of embryo. So from zygote to blastocyst or so. Okay. So it starts in late gametogenesis. And in the genome of every one of us, 15 to 30% of our genome, there are so-called imprinted zones. And the imprinted genes are clustered in these imprinting zones. Imprinting zones, imprinted zones, or imprinted areas, there are synonyms. Okay, so 15 to 30%, 1/3 of our genes are imprinted. Okay, in imprinting zones, in these parts of our genome which are imprinted, some genes are imprinted by men, some genes, not the same genes, are imprinted by women. There are different patterns of imprinting in male and female gametogenesis. And in every so-called imprinted gene, only one allele is really imprinted. Yes, so it is imprinted or by man or by woman. Okay, when it is when, for example, every gene has 2 alleles. Yes, when one allele is imprinted by, for example, man, the gene, the second allele is not imprinted by woman. Yes, if it is if one allele of another gene is imprinted by woman, it is not imprinted by man. So when one allele is imprinted by man, it is not imprinted and stays active in woman's or mother's gametogenesis, and inverse. Okay, like here on this picture, and here you can see a fragment of a chromosome in the future mother and future father, and late gametogenesis. Yes, and we can see this first gene, it is imprinted in paternal gametogenesis. So paternal allele is imprinting, it is inactive. Maternal allele is active. The second gene, it is imprinted in this it is methylated in maternal gametogenesis. Maternal allele is methylated, it is imprinted. So paternal allele must be active, and so on, so on. One allele must be always active. Yes. So if one allele is imprinted in father, future father, it is not the second allele is not imprinted in future mother, and inverse. In the somatic cells of a child, in which the complex of genes from mother and from father meet, we can see a constant pattern of imprinting in imprinting zones. Yes, like on this last picture. It is a stable feature which depends only on sex of a parent, not of sex of a child. Yes, so there is a pattern of imprinting of men, pattern of imprinting of women, and these which are not the same, totally not the same. Yes, they meet in zygote, and they repeat this pattern in all cells of the new organism. Yes, It is a stable pattern of imprinting. Like on the last picture, yes. When a gene is active in mother, it is not active in father. When it is active in father, it is not active in mother. 2 chromosomes in every somatic cell of a child, and in imprinting zones, it should be like this. When one allele is inactive, the second one must be active. One allele inactive, the second one is active, and so on. So, okay. Always one allele must be active, because if not, the gene, the whole gene is inactive. In imprinted areas, genes behave active or not, depending on the sex of a parent from whom a particular gene was inherited. Yes. In non-imprinted areas, genes behave independently of sex of a parent from whom they were inherited. So, in imprinted pattern, in imprinted areas. Just a moment. In imprinted, it was like this, yes. When it is a gene is imprinted in father, in chromosome originated from father, it is not imprinted in chromosome originated from mother, and so on, so on. Yes, it is inactive in father, it is active in mother. Okay, but in non-imprinting part of our genome, the genes behave independently on the sex of a parent. It is also somatic cell, chromosome on the left, chromosome from mother, on the right, chromosome from father, and we can see normally our genes should be active in non-imprinted zones. Yes, so we have this first 2 genes. They have 2 alleles which are active. Okay, the third gene, one allele which is active from a mother, and one allele which is inactive from a father, because, for example, of mutation, heterogeneous mutation of this gene. Yes. And the next gene, the fourth gene, is active. Both copies are active, both alleles are active, and the fifth gene is totally inactive. Both copies are inactive. Why? Because of maybe they are not methylated, they are not inactivated by imprinting. They may be inactivated by mutations, recessive mutations also. Okay, 2 recessive mutations. There are questions or comments? If not, we continue. In tight gametogenesis, there is a removing of parental imprinting. A removing of parental imprinting take place, yes, and this child adds of a new imprinting on these genes which are situated in imprinting zones. And this new imprinting depends on the sex of this child. Okay? So, in early gametogenesis, there is a removing of the imprint of a paternal parental, of the parental imprinting of genes. Yes, so all genes became active. Yes, this is on the left maternal chromosome, on the right paternal chromosome, and the methylation of all genes means that all genes are active during the early gametogenesis. Yes, and next, this child at its own imprinting. For example, this child is a girl, yes, and in her late gametogenesis, she add the female imprinting on the chromosomes, on the gene, on the chromosomes, yes. And what does it mean, maternal imprinting? On both chromosomes, the same imprinting, okay, the same pattern of imprinting. But these chromosomes will be separated during the formation of mature germinal cells. Okay, so it will be not that both these chromosomes will be in a zygote, but only one of these chromosomes. The second one will be from a partner of this girl. So it should have a totally different pattern of imprinting. In general, a pattern of imprinting is specific for species. For example, Homo sapiens, right? And imprinting zones are present in all autosomal chromosomes. Yes? And the region, which is situated on the long arm of chromosome 15, 15q11q13, is one of the most unstable regions of the human genome, which means in this case that all genes in this region are imprinted, have one allele which is imprinted. And here we can see this region, the special imprinted region. It is from Q11.2 to Q13. On the left, maternal chromosome 15. On the right, paternal chromosome 15. Only a small fragment of these chromosomes from Q11.2 to Q13, and in a part of this region we can see five genes, and in maternal chromosome these five genes are one of these five genes is active, other genes are inactive. This the first gene is active because it is not methylated. The genes from number 2 to number 5 are methylated, are imprinted, so they are not active. Okay, and in paternal chromosome, this first gene is inactive because it is normally in all men, it is inactivated by methylation. Other genes are active. So in every one of these 5 genes, one copy is active, one copy, which means allele, is inactive. Okay. And what is the mechanism of genome imprinting? In general, there are many theories of this mechanism. There are many facts also about this mechanism, but I should have 10 hours to explain you all the possibilities of a female organism and another organism. No, nothing. Yes. So it is a stable pattern of this genome of these parthenogenetic organisms, and this stable genome is transmitted from generation to generation. Okay, only female generation. There are many organisms now, even now, which are parthenogenetic, yes, but in general, this way of breed is not very practical. Why? Because there are not the exchange of genetic material, and um, this exchange allow for better living, allow for good health, even if environmental factors change. Yes, so it's better to have, for example, um, the crossing over or something like this type of mechanism than the same genetic material from one generation to the next generation, because environment uh, changes all the time. Yes, and we should adjust ourselves to this uh, these changes of environment. And parthenogenetic organisms uh, don't have this possibility to adjust, adapt fully to changes in environment, in, in the environment of the life in general. Okay, and the second mechanism, protection from double doses of genes. You can see that in this imprinting zone there are only one copy, always one copy active of a gene, one copy inactive. There is no situation of activity of both copies of a gene. Yes, for example, here. Oh, for example, here. Okay, no, it's not a good illustration. Here. Yes, always. Oh no, so it's not illustration. Sorry. Here, always one copy is inactive. Never both copies are active in these imprinting zones. The situation when both copies are active, it's not a healthy situation for this organism. It may cause genetic syndromes, it may cause genetic diseases. The question is why the nature gave us both copies if only one we need. Yes, if we need only one copy and two copies are harmful. I don't know. Maybe because of pathogenesis, maybe because of other mechanisms. Yes, you will see that double doses of genes is not good for the development of organism, that it may cause also genetic syndromes. And here you can see genetic material in parthenogenesis. Both chromosomes have the same imprint, have the same pattern of imprint, yes, and they have the double doses of many genes, yes, double active genes, which are red, and double inactive genes. So the imprinting caused the death of many parthenogenetic organisms in the evolution. The imprinting is a totally physiological mechanism, but it has an impact on disclosing, on revealing some microaberration syndromes, monogenic diseases, and neoplasm. When something appears, a change appear in imprinting, in mechanism of imprinting, or in this part of genome in which this imprinting should be, always the results are harmful for the organism. Always, as a result, we have genetic syndrome, or, for example, monogenic disease or neoplasm or so on. And now we'll talk about so-called uniparental disomy. Uniparental disomy, UPD, it's a totally different mechanism and process from imprinting. It is a pathological phenomenon, completely pathological phenomenon, not physiological as imprinting. Okay, and what does it mean, uniparental disomy? It means that two chromosomes in a chromosome pair, so-called homologous chromosomes, origin from the same parent. Yes, normal situation is that one of these chromosomes origins from one parent, the second one from the second parent. Yes, but here from the same parent. And we have at least two mechanisms of this uniparental disomy. The first one is heterodisomy. In this mechanism, we have two different chromosomes, but homologic chromosomes, yes, from the same pair of chromosomes, yes. Why? Because of meiotic non-disjunction. For example, you have two parents, one green parent, one red parent, and this green parent gives one chromosome of a pair of chromosomes, of this pair of chromosomes, to his or her germinal cell. But the red parent, because of non-disjunction of this pair of chromosomes, gives both chromosomes from this pair to germinal cell. Yes, and we have fertilization, and trisomic zygote appears. Yes, trisomic zygote, three chromosomes in a pair of chromosomes. It's not the very so-called pleasant situation for a zygote, because trisomic zygotes are in majority alethal. Yes, alethal is the stage of zygote or later on. Yes, even children trisomic for chromosome 21, they are not born from every trisomic zygote. Yes, most trisomic, most zygotes which are trisomic for chromosome 21 die during the first days of their development. Okay? So it's better for a zygote not to be trisomic. So this zygote may throughout one of these chromosomes from this trisomy. Okay, it may be one of the chromosomes of this red parent. It's the best situation because when one of the red chromosomes is expulsed from the cell, the zygote has only two chromosomes, one from one parent, the second one from the second parent. Yes, and this situation is normal. And the probability of expulsing one of chromosomes of this red parent is 2/3. Yes. But there is also a probability of expulsing this chromosome, only one chromosome from the green parent. Is 1/3, but sometimes this green chromosome is expulsed from a zygote. So a zygote became a non-trisomic, only disomic zygote, yes, with a very beautiful pair of chromosomes, but they originate from the same parent. They have the same imprinting, parental imprinting, and they are not identical, they are homological chromosomes. Yes, and now I can present you some example of complications of this situation. For example, we can, for example, call these chromosomes sex chromosomes. For example, this chromosome from green parent will be X chromosome, yes, and this chromosomes from the red parent, X and Y chromosome. This Y chromosome is fulfilled by the red stain, yes, and these empty chromosomes are X chromosomes. Okay, so we have trisomic zygote which have two chromosomes X and Y and one chromosome Y. One of this green chromosome X is expulsed, yes, and the zygote has only two chromosomes, Y and X and Y, inherited from this red parent. Red parent because he has one chromosome X and one chromosome Y. He is a father of this zygote, this organism. Yes. So, for example, this father has hemophilia or Duchenne muscular dystrophy or other or another X-linked disease. Okay, and because this child in the lower part of this design of this picture, because this child inherits both sex chromosomes from this father, this child inherits also this gene for hemophilia or Duchenne muscular dystrophy or so on. Okay, and in normal situation, this mechanism doesn't exist. The son cannot inherit X-linked disease directly from his father. Okay, there is no no direct transmission of sex-linked diseases from father to son. Okay, you know that all sons of father who has X-linked disease are healthy. Yes, but in the case of heterodisomy, this son may inherit X-linked disease directly from his father. We should, yes, but the question is if chromosome Y and X are homological chromosomes. In a certain sense, they are because they have the pseudo-autosomal regions which are homologous. Yes, but another example, for example, these chromosomes are autosomal chromosomes. Yes, and of course this is the same pair of chromosomes, and this green parent is not ill, is not a carrier of a recessive gene on this chromosome, but this red parent has recessive mutations of both homologous chromosomes. Yes, so this red parent is, he or she in this case has a recessive disease, because of two copies of recessive gene on both copy of these homologous chromosomes. Yes, so both chromosomes are changed, both genes are mutated, and here we can see the direct transmission of autosomal recessive disease to a child. In normal transmission, it should be that the children of this red parent may inherit only one recessive copy of gene, one copy of this recessive gene. So all children of this red parent are heterozygotic carriers of this recessive gene. Yes, they should not be ill. They will be carriers of recessive genes. Only carriers, not ill. Yes, and here, because of this heterodisomy, you can see that a child will be ill like his or her father, mother, like his or hers is red parent. Yes. So, summing up, the result of this heterodisomy maybe is non-Mendelian manifesting of recessive traits, not changes in Mendelian rules, but non-Mendelian manifesting, not inheriting of recessive traits. Transmission of hemophilia or Duchenne muscular dystrophy from father to son. There is one assumption that X and Y are homologic chromosomes. Yes. Or if these chromosomes are not sex chromosomes, autosomal chromosomes, imagine that they are autosomal chromosomes, abnormal transmission of autosomal recessive diseases. Yes. And the second type of uniparental disomy, so-called homodisomy or isodisomy. Two identical chromosomes. It's iso, the same. Yes, the same chromosomes. The problem is that the green parent lost his chromosome of this pair in the anaphase of meiosis. So green chromosome doesn't exist. And we have only monosomic zygote. Yes, here. And all autosomal monosomic zygotes are lethal. No chance to be alive. Okay, so this zygote should act quickly to rescue itself from monosomy. Okay, so the zygote replicates this chromosome. Yes, a new zygote has two chromosomes of this pair. The quantity of genetic material is normal, but they are identical chromosomes. And imagine the situation that this red parent has on this chromosome an autosomal recessive gene of a disease. Yes. So this child, which will be born from this zygote, will have this disease inherited from this parent. The green parent has no mutation of this gene. Okay, so non-Mendelian manifesting of recessive mutations. So in this, what else? Only one carrier, autosomal recessive carrier of parents, and the child which is here. Okay, because of duplication of a recessive gene with mutation. Duplication of the whole chromosome means also duplication of all genes on this chromosome. Okay, and what else? Independently of mechanism of UPD, if it whether it is homodisomy or heterodisomy, there is always a possibility of abnormal gene function, which depends on genome imprinting, because both chromosomes in these two situations have the same pattern of imprinting. Yes, so both alleles of a gene may be silenced, both alleles may be active. When both alleles are silenced, it means that the whole gene is inactive. Yes, so it means a disease. Both alleles active means that it is too much active material, active genetic material of this gene. And what does it mean? Also a disease because of imprinting abnormalities. Okay, so these two types of uniparental disomy cause the diseases which may result from abnormalities of imprinting, and also because of abnormalities in Mendelian manifesting of recessive traits. And what it can be, for example, maternal UPD, uniparental disomy, which is maternal, both chromosomes origin from mother, so both chromosomes have the same pattern of imprinting, and or both alleles are active or both alleles are inactive in imprinting zone in which only one allele of every gene should be active. Okay. And now I would like to present you the enigma, so-called enigma of the deletion of this 15q, 11q, 13 region. This is one deletion, okay? And the same deletion, in some cases, causes one phenotype, in other cases, totally different phenotype. For example, in some cases, it causes Prader-Willi syndrome, and in some, so-called Angelman or Angelman syndrome. Why? And you can see that these syndromes have totally different phenotypes. For example, Prader-Willi syndrome, light psychomotor retardation. Yes. Angelman syndrome, very heavy mental retardation. In Prader-Willi, no ataxia. In Angelman, cerebellar ataxia. In Prader-Willi, huge obesity, hyperphagy, hypogonadism, stereotypic behavior. In Angelman, no obesity, no hyperphagy. Yes. Sometimes hypogonadism, sometimes stereotypic behavior. In Prader-Willi syndrome, no frequent changes of mood. But in Angelman syndrome, very frequent changes of mood. Okay. Frequent bursts of wrath. Frequency of this syndrome is also different. Why is this big differences between two syndromes which are caused by the same cytogenetic change? Because it depends on the origin of chromosome in which occurs this deletion. When this deletion occurs on paternal chromosome 15, it causes Prader-Willi syndrome. If this deletion occurs on maternal chromosome 15, it causes Angelman syndrome. So there is no difference, between the region of deletion, between the quantity of deleted genetic material. There is only difference between the origin of chromosome 15. from father, it means that Prader-Willi syndrome will appear in a child, or from mother, chromosome 15 with deletion. If it is from mother, Angelman syndrome will appear. Okay? You understand? And the difference between these children, on the left, Prader-Willi syndrome, on the right, Angelman syndrome. And next photo, Prader-Willi syndrome, with huge obesity, hypogonadism, and so on. Okay? And on the next our lecture, I will continue about on Prader-Willi syndrome and Angelman syndrome. Do you have, because we should finish, I think, yes? Yes. Okay. Yes. Yeah. And do you have any questions, comments?
No, Professor.
No, Professor.
No questions, no comments. Okay,
Thank
thank you. We will meet in, I think, 2 weeks. Okay. And in 1 week, you will meet with Professor Pasińska. Okay. Not today, but in a week. Okay. Okay.